Hello, Bio178, and welcome to our week 2 lab. Sorry, my neighbor's gardener just started blowing leaves, bad timing. So welcome to week 2 lab, and what we're going to do in lab this week is do the first hands-on processing of your samples for our semester-long group projects. So what we'll do in this lab is get the samples and prepare them for DNA extraction. So all the protocols are listed in this module. And one thing to note is that there are 2 different protocols. So depending on which group project you picked and signed up for, people are working with 2 different organisms, either the black turban snail, Tegula funnebralis, or the red abalone, Haliotis rufescens. So I provided protocols for each of those because they're slightly different depending on which organism you're using. So when you click next in this module, make sure you're looking at the written protocol for whichever project you signed up for. That's the first thing to note. So make sure you review the protocol, print it, bring it with you to lab. And the other thing that I'll note for this week's lab is that we also have a short lab report associated with this week's lab. So I say short, it's 5 questions. It shouldn't take you very long at all, but what I do recommend is looking at that ahead of time, and I would also maybe recommend bringing that with you to lab as well, because one of the things you're going to need to include in your lab report is some data and information about the samples that you processed in lab. So it's always good to know ahead of time what information you need to write down and record. So those are the main things that are included in this module that you'll need to review and bring with you: the written lab protocol and the lab report as well, so you know what information to record. Let me know if you have any questions. Otherwise, this should be a fun, exciting lab for us to get started with our group projects and get some hands-on experience with these samples. So I will see you soon.